Welcome back, guys. Today we are going to discuss this important topic in biochemistry, that is our glycogen metabolism. Glycogen metabolism. Okay, so let's get started with it. First, we are going to see this glycogen. Okay. We know the glycogen is the storage form of the glucose. Glucose is transformed to glycogen and is stored in the animal body. So glycogen is a branched polymer of alpha D glucose. Okay, the branched polymer of alpha D glucose. Okay, now the linkages in glycogen. Linkages alpha 1, 4 in the linear form. Okay, and alpha 1, 6 linkage in the branching form, or we can also say the branching is at alpha 1, 6. Okay, the linkages are in the alpha 1/4 form. Okay, so it's a branched polymer alpha D glucose. The branching is at alpha 1,6 position. Okay, now the storage of glycogen. Okay, the storage of glycogen. Now the storage of glycogen, it is stored as cytoplasmic granules. Cytoplasmic granules. Okay, so it is this molecule called alpha rosette. Okay, alpha rosette. Let me write this again. ROSETT, alpha rosette. Okay, this contains 20 to 40 beta particles. Okay, 20 to 40 beta particles, and remember, a single beta particle is 21 nanometer in diameter. 21 nanometer in diameter, a single beta particle. Okay, so this single beta particle, okay, has 55,000 glucose units. 55,000 glucose units. Okay, and it has 2,000 non-reducing ends. 2,000 non-reducing ends. Single beta particle has 55,000 glucose units. So this is the how glycogen is stored. Now the main question arises, why glucose is stored as glycogen? The obvious question arises, why glucose is stored as glycogen? So we have some reasons. First reason being, it is compact in nature. Second, it has multiple reducing ends, or exactly to be multiple non-reducing ends are available. Multiple non-reducing ends are, so glucose can be released faster. Okay, I discussed the non-reducing ends here. So there are multiple non-reducing ends that aids in the storage of glucose as glycogen. Okay, the third amount is that glycogen does not attract water molecules. Attract water molecules. Okay, to cause cell lysis. Glycogen does not attract water to cause cell lysis, so higher amount of glycogen can be stored in lesser concentration as compared to glucose. So higher amount of glycogen can be stored in lesser concentration as it does not attract water molecules, and no water molecules can cause cell lysis. So this is third reason, one of the most important reason why glucose is stored as glycogen. Okay, this was all about storage and structure of glycogen. Now we are moving on to the main part, this metabolism. Okay, so first is the synthesis of glycogen. Obviously we know this is glycogenesis. Okay, glycogenesis. This glycogenesis occurs, remember, in fed state, when food is present in the stomach in our body. Okay, fed state 4 to 6 hours after food, or postprandial state. We have pre-prandial state before food and postprandial state after food. So 4 to 6 hours after food occurs in fed state, wherein organelle of cytoplasm. Cytoplasm is the site of glycogenesis, synthesis of glycogen. Okay, and remember, in the fed state, high insulin glucagon ratio. Very common thing to understand. Whenever there is food in the body, in the stomach, insulin will be released. Insulin is anti-hypoglycemic, so glucose will be converted to glycogen. So this glycogen storage or formation of glycogen in high insulin glucagon ratio in fed state. Okay, in cytoplasm. Okay, now the organs are, remember, liver and muscle. Okay, liver and muscle. This muscle, one fact remember here, that it comprises nearly 1 to 2% total weight of glycogen. Okay, and liver constitute 10% by weight. Okay, 1 to 2% in muscle and liver 10%. So this liver and muscle takes part in the glycogenesis process. Okay, now we will go see the steps of glycogenesis, formation of synthesis of glycogen. Okay, first step, remember, synthesis of UDP glucose. Okay, let's concentrate here, synthesis of UDP glucose. So what happens here is, here we have the entry product is glucose. Glucose is being converted to the storage form glycogen. Entry product is glucose. It's forming glucose 6-phosphate. Very simple, we know the enzyme hexokinase. Okay, nothing fancy here. So glucose 6-phosphate is getting converted to, remember, glucose 1-phosphate, and the enzyme here is phosphoglucomutase enzyme. Phosphate is removed from 6th position to 1st position, so phosphoglucomutase. After glucose 1-phosphate is formed, it is combined with UTP. Okay, what is this UTP? Uridine triphosphate, like ATP, adenine triphosphate. Adenine is a nucleotide, and uridine is a nucleotide form, triphosphate, uridine triphosphate. Okay, in this process, this phosphate, this 1-phosphate in the first position in the glucose 1-phosphate is removed. This is the important. Okay, combination of the UTP phosphate is removed from glucose 1 phosphate. Okay, and finally UDP glucose is formed. UDP glucose here is formed. There is release of a EPI, inorganic diphosphate, is released here. Okay, and the all important enzyme here is UDP glucose what pyrophosphorylase. UDP glucose pyrophosphorylase, the enzyme here. Okay, and the important thing to note here, that here the hexokinase, we know it utilizes ATP to form ADP. Okay, hexokinase utilizes enzyme to glucose 1 phosphate. Combination of the UTP, the phosphate is removed. Formation of UDP in presence of UDP glucose pyrophosphorylase, inorganic diphosphate is also removed. So first part of UDP glucose formation is over. Next is our glycogen synthase enzyme step. Glycogen synthase enzyme. So first here I am only saying this is the rate limiting enzyme. It's the rate limiting enzyme, glycogen synthase. Okay, so first we have this structure here. Okay, these are a bit complicated steps, so listen carefully. The structure is called a glycogenin. Generation of glycogen from this substance, so called glycogenin. This is a primer. Okay, remember this is a primer. Okay, now we have here, what do we have here? Residues getting added. What residues? 7 to 8 residues of glucose are added. Okay, from where? From UDP glucose monopolymer. It's multiple N UDP glucose that was formed here. It's formed polymer UDP glucose multiple times. From this 7 to 8 residues of glucose are added to glycogenin, which is a primer. Obviously, in the presence of our enzyme, our rate limiting enzyme, glycogen synthase. Very simple. Upon this action of glycogen synthase, the structure is formed. Okay, this is our glycogenin originally was formed. After addition of 7 to 8 residues, 2, 3, this structure is formed. Okay, and here UDP glucose polymer is being converted to UDP glucose monomer. Okay, this structure is formed. We don't usually have a name for this. Okay, so this structure is formed here. Okay, this is the formation usage of glycogen synthase enzyme. Now after this step, the third step is formation of branches. We know branching of glycogen is important at alpha 1/6 position, formation of branches. Okay, so first the branching enzyme. Branching enzyme is alpha 1,4,1,6 glucan transferase. Alpha 1,4,1,6 glucan transferase. So the previous molecule here was formed by the glycogen synthase. Okay, I'm going to draw this here. Okay, as the branching is at, we know, 1/6 position, so it's 1, 2, 3, 4, 5, 6 glucose residues in this structure are cut and removed. 1, 2, 3, 4, 5, 6 glucose structures are removed. Okay, this branching enzyme acts in this removed, and what is formed here after removal, it gets reintegrated to a different position. Okay, so it gets reintegrated here. Okay, 1, 2, 3, 4, 5, 6 gets reintegrated here. So here it comes alpha 1, 6 glycosidic linkage. Okay, it may seem a bit different, a bit weird, but this is how books have illustrated and how this process takes place. 6 glucose residues are cut, that is branching enzyme removed and reinstated here to form alpha 1,6 glycosidic linkage. And what is this? Our glycogen monomer is formed. Okay, so first was UDP glucose formation, second was glycogen synthase, removal of residues and monomerization of UDP glucose, and then this formation of branches and formation of glycogen. Very easy steps, 1 to 2 times revision will do it. This was glycogen synthesis. Now we have the next all important glycogenolysis. Glycogenolysis, breakdown of the formed glycogen. Okay, and here we'll start with the different stages of fasting. Okay, this glycogenolysis occurs in fasting stage. Okay, 4 to 16 hours after food. This fasting stage after 4 to 16 hours is called early fasting. The previous glucogenesis video, we have seen that glucogenesis takes place in fasting stage, which was 16 to 48 hours after food. Before that, this glycogenolysis takes place in the early fasting stage. Okay, these are post absorptive stage or post absorptive stage. And this glycogenolysis obviously takes place in liver. Okay, this is our hepatic glycogenolysis. Which is as the source of blood glucose level. Our blood glucose level in the early fasting stage is maintained by this hepatic glycogenolysis. It is in liver the glycogenolysis is taking place. And here, as in the fasting stage, the hormone acting here is glucagon, not insulin, because the food is not present here. Food is being broken down, so insulin level is low. So in low insulin glucagon ratio, these levels of insulin glucagon are very important for understanding. Here the main hormone, main hormone acting here is glucagon in low insulin glucagon ratio. Okay, also takes place in skeletal muscle, along with liver skeletal muscle. Organelles important: cytoplasm, lysosomes, and smooth endoplasmic reticulum in liver. Harrison specifically mentions this smooth endoplasmic reticulum in our liver. Okay, others say cytoplasm, and lysosomes also has a function in glycogenolysis. Okay, so Now we will see the steps. Steps of glycogenolysis. Glycogen was found. Now it's need to be broken to supply the blood glucose. Okay, in early fasting stage. So the first enzyme, role of enzyme here is glycogen phosphorylase. Glycogen phosphorylase. Okay, first here I am telling it is the rate limiting enzyme. Okay, it's a rate limiting enzyme. Okay, what is the function of this glycogen phosphorylase? We have glycogen molecule will be drawing here. Okay, with our alpha 1,6 linkage. Okay, alpha 1,6 linkage here. Okay, this is the glycogen molecule. And here some more extensions will be there. Okay, so glycogen polymer. So glycogen. Okay, now glycogen phosphorylase acts here. What it will do? See here. It will remove the end 4 glucose molecules. Okay, end 4 glucose molecules. Remember, glycogen phosphorylase here requires PLP. Okay, PLP is required here, and an inorganic phosphate is used to form side product glucose 1 phosphate. Okay, glucose 1 phosphate is here, is formed. We will learn about this in the next subsequent steps. What is the use of this? Remember, this requires PLP. Inorganic phosphate is used. Remember, these are glucose residues. Okay, so as the glucose residues are removed, glucose 1 phosphate is formed. And in the product, what is found? Or limit dextran. Okay, limit dextran is formed by the action of glycogen phosphorylase on glycogen polymer. In the byproduct, glucose 1 phosphate is formed. This is the first step. Glycogen phosphorylase is the rate limiting enzyme in glycogenolysis, and there the rate limiting enzyme was glycogen synthase. Okay, moving on to the next step. In step 2, okay, there is removal of branches. There was formation of branches in the glycogen synthesis. In the third step here, removal of branches, and this is done by our debranching enzyme. Debranching enzyme. So this debranching enzyme is a, remember, bifunctional enzyme. It's a bifunctional enzyme. Okay, this has two functions. Remember, first being alpha 1,4 glucan transferase. Okay, this is one enzyme function. Another is alpha 1,6 glucosidase. Alpha 1,4 glucan transferase and alpha 1,6 glucosidase. These two are bifunctional enzymes of this debranching enzyme. Okay, now we have our this structure here, limit dextran. So we will continue with this limit dextran. Limit dextran enters into the cycle. Okay, the glucose residues are here. Okay, now these 4 glucose molecules are remaining here. Out of these 4 glucose molecules, here these 3 glucose molecules, okay, are cut and relocated. These 3 glucose molecules at the end are cut and relocated. Relocated to where? Here, in the other branch itself. Okay, and this is the function of the first enzyme, alpha 1,4 glucan transferase. Very simple. Glucose residues are cut and relocated to here. So what will be formed now? These are original glycogen molecule from glucose synthesis. Okay, glucose residues are joined. The last 3 glucose are joined, leaving with 1 glucose residue here. Okay, this was due to activity of 1414 glucan transferase. Okay, now here action of the second enzyme. Okay, alpha 16 glucosidase. This one, this single glucose molecule cut and removed. So the single glucose molecules will lead to free glucose is being released. Free glucose molecule is being released here. Alpha 16 glucosidase action enzyme. The final product is formed. So a debranching enzyme has completed its purpose. Okay, the product is formed. No specific names are given here because this is the penultimate state and the ultimate steps are remaining out there. This is the product form and no branching here is formed. There are no branches here. So that's why this was called debranching enzyme. So bifunctional enzyme, removal of branches. These two enzyme function, alpha 1414 glucan transferase and alpha 16 glucosidase. Okay, and finally, most importantly, free glucose is being released here. Okay, so in the first step, I have said to emphasize on this glucose 1-phosphate with the addition of inorganic phosphate. As this glucose were being removed, glucose 1-phosphate is formed. Now this glucose 1-phosphate is to be converted to glucose. That is our ultimate step in the glycogenolysis step. We are breaking down glycogen to glucose. The blood needs glucose in the low insulin glucagon ratio in the fasting state. So let's see the conversion of glucose 1-phosphate to glucose. Okay, it's a bit complicated step here, not too much complicated because 2 organelles are involved here, so 2 different processes are involved here. So stay with me. Okay, first we are going to see in liver. Okay. In liver, why I am saying 2 different organelles are 2 different processes? Because some organelles here and organs lack some enzymes required for these conversions. We'll see in liver. Here we have glucose 1-phosphate and this is simply being converted to glucose 6-phosphate by the same enzyme, phosphoglucomutase enzyme, or simply mutase enzyme. And this glucose 6-phosphate by the action of glucose 6-phosphatase. In which process? In gluconeogenesis is converted to or 6-carbon glucose, which is free and released. It enters this glucose 6-phosphate after formation, enters the gluconeogenetic pathway. The action of glucose 6-phosphatase, 6-carbon glucose is formed, which is free and released. Now in muscle, in our skeletal muscle. Okay, this is our skeletal muscle. Okay, in muscle we have our glucose 1 phosphate. We intend to convert it into glucose. Okay, so it is converted to glucose 6 phosphate because this muscle have our phosphoglucose mutase enzyme. Same mutase enzyme. Okay. The problem here is this step of formation of glucose is not possible because glucose 6 phosphatase is not present in muscle. Is not present in muscle. This is the dilemma here. This step does not take place. So glucose is not found. So we cannot allow this glucose 6 phosphate to stay there. There is some alternate pathway. Okay, this glucose 6 phosphate, very simple, enters anaerobic glycolytic pathway. Glycolytic pathway. Okay, and finally pyruvate is formed. Pyruvate is formed, which will ultimately lead to an anaerobic glycolytic lactate. Okay, and very simple ATP is released. We have discussed total of 2 ATPs gained here, 2 ATPs released here. Okay, lactate is formed, and here the specialty is 3 ATPs are released instead of 2. 3 ATPs are released. Why? As hexokinase is not involved here. So hexokinase is not involved here. So 1 ATP that was being consumed is not being consumed. So 4 are produced. Only 1 is being utilized. So this will lead to 3 ATPs being released, and as hexokinase is not being involved here. Okay, this is the peculiarity of this step, conversion of formation of lactate in muscle from glucose 1 phosphate. Okay, remember, in liver glucose is formed and released. In muscle, closing glucose 6 phosphate is not present, so anaerobic glycolysis takes place, lactate is formed, and 3 ATPs are totally produced. This was all about our glycogenolysis. First, glycogen phosphorylase reacting enzyme acts to form limit dextran. This limit dextran is an important molecule and a harmful molecule. If it accumulates, it will cause some glycogen storage disorders. We will discuss in further sections. Then there is removal of branches, the action of debranching enzyme here. And finally, free glucose being released. In the third step, glucose 1 phosphate need to be converted to glucose. The process is different in liver and in skeletal muscle. Okay. Now I'm going to see some regulation of glycogen metabolism. Hormonal regulation, the allosteric regulation, all will be seen here. So first is our hormonal regulation. Hormonal regulation. Okay. Hormonal regulation depends on dietary status. Dietary status is depending on hormonal regulation. So obviously in fed state we have insulin. In fasting state or during exercise, okay, fasting state or during exercise, we have glucagon. We have glucagon along with epinephrine. Epinephrine hormone is also taking part in this fasting state or exercise state. in glycogen metabolism. We'll see how. Okay, so we have here our cell membrane. Okay, here we have is glucagon. Okay, so glucagon, and here we have our G protein coupled receptor molecule, here GPCR molecule. Okay, now we see this action, glucagon and epinephrine action. We have our glucagon and epinephrine both. So this we have here our glucagon, this GPCR, and also our epinephrine. Okay, what happens next is, after they combine here glucagon and epinephrine, this G protein goes inside the cell membrane. Okay, this is our G protein, and with the help of GTP, here it's our adenyl cyclase molecule. Adenyl cyclase molecule, it is activated here. G protein with the help of GTP, adenyl cyclase is activated here, and so its level increases. Okay, upon this activation, this will lead to conversion of ATP to CAMP. Okay, which will lead to conversion of 5-AMP. Okay, and this CMP, this adenyl cyclase, will form CAMP dependent protein kinase A. CAMP dependent protein kinase A. Protein kinase A. This is the single most important product formed here. CMP dependent protein kinase A is formed. Okay, what is this role here? This CMP dependent protein kinase A. Okay, here it's a phosphorylating enzyme. Here it's a phosphorylating compound. Okay, we are seeing this, the action of glucagon and epinephrine here in the fasting state. So we know in fasting state glycogenolysis takes place. So glycogen synthase is inactivated. Very simple. And glycogen phosphorylase is activated. Phosphorylase is activated. So glycogen synthesis does not take place, glycogenolysis takes place. Both of these are getting phosphorylated. Both these enzymes are phosphorylated. Upon this phosphorylation, this synthase enzyme is being inactivated, glycogen phosphorylase enzyme is being activated. Who does this phosphorylation? CMP dependent protein kinase A. Okay, by the action of which two hormones? Glucagon, epinephrine. Okay, so here glycogenolysis takes place and glycogen synthesis does not take place. Okay, now in fed state, we have seen in the fasting and exercise state. In fed state, insulin activates this important enzyme phosphodiesterase. You must have come across this enzyme numerous times before. Phosphodiesterase. This enzyme actually converts cyclic AMP to 5-AMP. Okay, and this level reduced. Okay, as this level is reduced, this molecule is not formed. CMP dependent protein kinase A is not formed. So there is no phosphorylation. So glycogen synthase and phosphorylase are both dephosphorylated. Okay, and insulin is important in this step. Okay, ultimately in fed state glycogen synthesis becomes active and glycogen synthesis takes place, and ultimately phosphorylase is inactivated, glycogenolysis is inhibited. And who is responsible for all this? Insulin. Who was responsible for all this? Glucagon and epinephrine. Here the key concept is CMP dependent protein kinase A, and the two hormones, insulin, glucagon and epinephrine. Okay, so this was the hormonal regulation of glycogen metabolism. Okay, next is we have allosteric regulation. Okay, so here we have glycogen, here we have glucose 1 phosphate, and they are being interchangeably found here. Okay, first glycogen to glucose 1 phosphate, this is glycogenolysis. Okay, genolysis. Here ATP is inhibitor. Okay, ATP is our inhibitor here. Okay, glucose is inhibitor, and glucose 6 phosphate, remember, inhibitor. Okay, ATP is found, product is inhibitor, glucose is being released, glucose is a product, is being formed, is also an inhibitor, intermediate glucose 6 phosphate is found, is also an inhibitor. This is glycogenolysis. Okay, and here we have glycogenesis. Okay, from glucose to glycogen, and here G6 phosphate is activator. Here G6 phosphate is an inhibitor, G6 phosphate is an activator. This is the difference. Remember, this takes place in liver. Okay, now we'll see in what happens in skeletal muscle. In skeletal muscle, okay, again glycogen to glucose 1 phosphate, this takes place in glycogenolysis, this takes place in glycogenesis. Glycogenolysis. Okay, so here first of all in muscle, calcium, it's an activator of glycogenolysis. Okay, AMP is an activator. This two needs to be remembered. Okay, then our ATP is an inhibitor, and glucose 6 phosphate inhibitor. Okay, calcium and AMP are activator of glycogenolysis in skeletal muscle. Here in glucogenesis, this is phosphate is an activator. Now the point to noted here in skeletal muscle that glucose is not produced in muscle. We have seen that in the third step of glycogenolysis due to absence of glucose 6-phosphatase enzyme. So glucose is not produced in muscle. So no effect in allosteric regulation in muscles. Okay, as glucose is not produced in the muscles, so there is no effect in allosteric regulation in muscles. Remember, in muscles, in glycogenolysis, calcium and AMP is the activators. The rest others are usual inhibitors. Also, in glycogenesis, glucose 6-phosphate is the activator. So this was all about our glycogen metabolism. First we have seen the structure, the storage, then the glycogen synthesis, glycogenesis, and then the glycogenolysis, the destruction, how differently it acts in liver and muscle, then our hormonal regulation, action of insulin, epinephrine and glucagon, and then our allosteric regulation, also how it is different in both liver and muscles. Remember, the difference in muscles is that first is there is no glucose 6-phosphatase enzyme, and second, there is no action of hexokinase. Hexokinase does not act. Okay, glucose is not formed in muscle, instead lactate is finally formed in glycogen destruction or glycogenolysis. So this was all about glycogen metabolism. Please do revise them, guys. Thanks for watching. I will catch you in the next one. Good luck.